Hello everyone, welcome back to my channel. This video is the free class video for STPM Biology Semester 2 Chapter 11, Reproduction, Development and Growth. So this video is special for those who taken STPM Biology and in Semester 2. So let us see. Based on the MPM syllabus, this is the learning outcome that you require to know that must be come out in your examination. So in this chapter, there are 4 main big topics that you need to learn. The first topic is sexual reproduction in humans. So including what you already learned in SPM. So spermatogenesis, oogenesis, pressure of sperm, menstruation, fertilisation, implantation, all that you already learned. And also, there are also the new things that you need to know lah. Of course, the second topic is implant. So you also need to learn about the sexual reproduction in flowering plant. So including double fertilization, embryonic development, and there are the new topics which is the tissue culture and grafting in flowering plants. So this is a new topic that added in the new syllabus. And then the third topic is the seed germination topic. And then the final topic is the growth topic. So this chapter is a long chapter that we made, including the 4 big topics.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
So first of all, we go through the human sexual organs. So actually, this topic, I think I don't need to talk much about this lah, because I think including your Form 3 science, basic science, you already learn. SPM you also learn. So this is a anatomy structure of male sexual organs. So in STPM, normally they will not come out this lah. They will not come out, you label this, what is this, they already not come out, because in your syllabus is to want to know how the sperm is produced and ejected to the female sexual organ during the sexual intercourse lah. So this is the basic structure. You also need to know as well, because how the sperm is transmitted, you need to know lah. So first of all, normally your sperm is produced in the testis at here, right? In male, we know that sperm is produced in the testis. However, in biology scientifically, you need to mention scientifically, not just say testis, because testis have many structures. You can see, including epidermis, and also, of course, testis, and also including the scrotum. So normally, your sperm is formed by spermatogenesis in the seminiferous tubule. That one is you will see in later. I will focus on it during spermatogenesis. So it is in the produced in the testes through the spermatogenesis process. After that, your sperm will be passed into the coil duct of the epidermis at here. It will store at here for maturation about 3 weeks like that lah. About 3 weeks. So after your sperm is already produced in the testes, it will store in the epidermis first for the maturation process, and after during the sexual intercourse between male and female, meaning that they will have the ejaculation process, meaning that your penis will become more bigger and longer compared to you see like this. So during ejaculation, your sperm will starting transport from this epidermis and through the penis by the one tube we call as vas deferens. Here you can see all this. It will transported through this tube we call as vas deferens until it reach the behind of the urinary bladder at here. This is a urinary bladder of male, and it will until it locate at here lah, seminal vesicle. So it will reach the seminal vesicle. So what is the seminal vesicle do? So normally seminal vesicle is to secrete the semen. You need to know sperm is the cell. So how the sperm can be transported from the penis to the female sexual organ? They need a water. They require a medium to transport easily. So that seminal vesicle, prostate gland, and also bulbourethral gland, both of three of them will secrete the semen. But however, seminal vesicle play the major role to secrete the semen, so that the sperm can be transported easily from the male sexual organ into the female sexual organ during the sexual intercourse process. So normally the seminal vesicle will secrete the semen, which is very thick fluid, yellowish fluid, and also alkaline fluid like that. And also your prostate gland, this is the sexual spatial structure that containing in the male sexual organ. Female does not have this kind of structure. Prostate gland will also secrete the semen directly into the urethra, which is milky and thin. So seminal vesicle play the role to secrete thick semen, but this prostate gland is play the role to secrete thin semen. After that, you will see there are another gland we call as bulbourethral gland will secrete another clear mucus to neutralize any acidic urine remaining in urethra. Because you need to know that in male sexual organ, the sperm coming out from the male part is similar as the urine. Urethra, the sperm will also go through the urethra. Similarly, urine also go through the urethra. So before the ejaculation process, they need to make sure that in the urine, in the urethra, there are no any acidic urine adhere. Because sometimes your urine is acidic, so they need to make sure the urethra is neutral. Because if it is too acidic, meaning that when the sperm is passed through here, mostly your sperm will die. Because it is acidic condition, mostly your sperm will die. So they need to prevent of this gland, bulbourethral gland, will start to secrete the mucus, which can neutralize the acidic urine that remaining in the urethra. After that, your sperm will be transported within the semen, will enter through the urethra, through the ejaculatory duct, here, then to your penis. And after that, this fluid, fluid that containing sperm will be released out from the penis during the sexual intercourse. So your penis is normally is a electrical compulsory organ that delivery the sperm into the female sexual organ sexual organ. So normally that's why that penis is very important to the male one, because if the male that does not have penis, meaning that the sperm cannot be produced cannot be released out from the male part this is like that So this is a passage of the sperm from the testis into the penis during the sexual intercourse. Once they already after the ejaculation, once the penis sperm is released from the penis, it will enter the vagina, of course. It will enter the vagina at here. And then after that, your sperm will swim through the uterine wall, uterine, and it will swim through the oviduct. And at here is the fertilization process. Here. Is part right. Oviduct, or another name in your SPM is fallopian tube. Fallopian tube also acceptable. So at here is your fertilization. After that, you will undergo series of cleavage, and then through the implantation, and then undergo the implantation, then you will develop the fetus. So overall, this is how the human is formed, how the baby is formed. So in this topic, I am focusing only the passage of sperm from the male into the female. Okay, so that's all about this topic. So next, we go through the spermatogenesis here. So spermatogenesis is the process to produce sperm in the testes. So actually this topic you already learned in SPM So just a revision only. So this is the whole process of the spermatogenesis. I think you are very familiar with this diagram or not already, right? So spermatogenesis is more easier compared to oogenesis because it is directly processed. Not like the oogenesis, because oogenesis have more tricky part. Later you already know lah. Yeah. So normally spermatogenesis is produced in testes, right? So inside your testes, you can see there are many seminiferous tubules at here. There are many coiled seminiferous tubules that are that pack in the testes. So actually your spermatogenesis is occur in the seminiferous tubule. So when you enlarge, you will see this kind of structure, this kind of the structure in the seminiferous tubule, and all this process is occur inside it here. So first of all, you will have the primordial germ cell in the embryo stage. During the embryo stage, they have the primordial germ cell, and then after they will undergo the mitosis process to form mitosis process to form the spermatogonial stem cell. So this is during the embryo stage, during the baby stage. So it will form the stem cell first here. This is the very, very early initial stage. So it is a stem cell, meaning that this cell can be directly can be multiplied along the lifespan. So in male, if you are very healthy, meaning that this stem cell can be multiplied along your lifespan until you die. So once the spermatogonial stem cell undergo the mitosis process, they will form the spermatogonium here. And then at the same time, they also undergo mitosis process to form primary spermatocyte. So this is the final mitosis process. After that, they will undergo meiosis process. Not mitosis again. It will undergo meiosis 1, which is to form the haploid secondary spermatocyte. All of this stage is diploid. Meaning that the chromosome number still 46. Until here, they become 23 already, become haploid. And then finally, they will undergo meiosis 2 to form the spermatic, early spermatic. So this is still not the sperm cell yet. This is just a spermatic cell, which is not mature yet. They will undergo the differentiation and maturation to form the sperm cell. So this is the oral process of the spermatogenesis. So initially, they will undergo 3 mitosis process. If you want to more easy, actually only 2 lah, because the first one is in the embryo stage one. After the baby is already produced, you already in baby stage, then they will start from here, spermatogonial stem cell. They will have the spermatogonial stem cell. And when you reach the puberty age, they were starting to do the mitosis process to produce the sperm. So that's all about the spermatogenesis. So you can see this diagram. This is the hormonal control of in the spermatogenesis lah. So actually, in male also containing FSH, follicle stimulating hormone, and luteinizing hormone, similar like female, but the concentration is very low. Not like the female. Female has the high concentration, but in male, your concentration is very low. But they still play the role in controlling the process of spermatogenesis. So normally, FSH, these follicle stimulating hormone, they will stimulate this Stolly cell that located within the seminiferous tubule to nourish the development of the sperm. So this is one of the cell that can play the major role in the development of the sperm in the seminiferous tubule. And then there will also another hormone we call as luteinizing hormone cause the LEE ZHI cell to produce the testosterone. So this is a hormone, a male hormone, right? Testosterone, which promote the spermatogenesis in the tubule here. So this is This is the role of the FSH and LH. However, if testosterone is very high, it will provide the negative feedback mechanism. If the testosterone is very high, of course, similar at here. If the storage cell are too high, they will secrete the hormone inhibin, which also will produce the negative feedback, meaning that they will stop the spermatogenesis. So this is a role of the FSH and LH in the spermatogenesis regulation process. Okay, so this is a sperm structure. I think I don't need talk much. So they containing three major parts lah. The first part is your head. The second part is the middle space, is like the body part, and then the final part is your tail. So this tail is play major role for swimming in the fluid, in the semen, so that it can swim from the vagina into the fallopian tube for the fertilization. So they also the head. So this is a nucleus. So this nucleus is very important for the fusion of the another nucleus from the secondary oocyte in the female one to produce the human diploid cell diploid zygote. Okay, so this mitochondria is provide the energy so that the sperm have energy to swim in. So this is a basic sperm structure. Next, we go through the oogenesis. So oogenesis is quite complicated compared to male. So you see here, so similarly, oogenesis is for the female one. Then development the egg. We call as the egg. In general terms, we call it as egg. But for your examination, please don't write the egg. It is to produce the mature oocyte in the ovary. So oogenesis is happening at here, meaning that it is happening at here. It is happening at here. Okay. So in actually is similar. The initial stage actually is very similar to the spermatogenesis. But which the female embryo, the primordial germ cell will also undergoes the mitosis process to form the diploid or orgonium first. So this is a orgonium, and then after that they will undergo mitosis process to the primary oocyte. And this is the process that happening in embryo stage. So different from the spermatogenesis, formation of the spermatocyte and spermatogonium, this one is not happening in embryo stage. This one is happening when the male reach the puberty stage. In embryo stage only happen at here. Primordial germ cell in the embryo undergo mitosis to form spermatogonial stem cell. Only happening at here. After that, after the spermatogonial stem cell, all this process is happening when the male reach the puberty age. But for oogenesis, the first three process, the primordial germ cell, female primordial germ cell under mitosis to form orgonium, and then it will also undergo mitosis to form primary oocyte. These three process two process happen in the embryo stage. So this make the female have the limited primary oocyte number, because in embryo state they already fixed. All the number is already fixed. For example, if the female have 1,000 primary oocyte, then only 1,000. They cannot be multiplied again. Not like the male, because male have stem cell. They have spermatogonial stem cell, so they can able to multiple more and more similar stem cell in the seminiferous tubule. But for female, they have limited number. If entire span in embryo stage, they only produce 1,000 primary oocyte, then only 1,000 primary oocyte. They already cannot produce in large number already. So after that, they will form the zygote, your baby lah. Baby is already formed. Is already formed. But then the process is stopped. No process. All the process is stopped until the female reach the puberty age. So normally female for now puberty age normally is 12 years old. 12 years old, 13 years old, even earlier 11 years old. Some of the girl already start the puberty. So then start the puberty age, they will start to complete the meiosis 1. Because you can see here, during the primary oocyte, it present at birth, and it will arrested in the process of meiosis 1. So during the embryo stage, okay, they will already produce embryonic oocyte. After then this girl is already birth, at the birth stage, they will undergo meiosis one, but they stop at the prophase one only. After when they reach the prophase one, they stop. They will not continue to the metaphase, anaphase, and telophase. No. So they will stop at the prophase one. Once they already reach the puberty age, they will complete the meiosis one and start the meiosis two. And once they already complete the meiosis one, of course they will produce two type of cell, right? They will produce two type of cell, but only one of it is called as secondary oocyte. The another one is we call as first polar body. This one is useless. They are not the egg, they are not the secondary oocyte. They will be denatured from the denature lah in the future. So they are only secondary oocyte. Okay. After that, the secondary oocyte, they were starting to go through the meiosis 2, but they will stop at metaphase 2, meaning that during the secondary oocyte, they will undergo prophase 1, prophase 2, prophase 2, and then metaphase 2. Once it complete metaphase 2, it stop it, arrested at there. So anaphase 2 and telophase 2 not be started. So when this anaphase 2 and telophase 2 continue, then they will continue once after ovulation process and fertilization or process occur. Then they will complete the meiosis 2. But if there are no fertilization, ovulation only occur, but there are no fertilization process, meaning that the meiosis 2 is not completed. And this secondary oocyte will be released out through the maturation process. Maturation process. This is the girl every month will go through lah. So later we will see menstruation. So this is the secondary oocyte. So your secondary oocyte is released from the ovary through the ovulation process, and it will reach the fallopian tube. And once there are fertilization process, it will complete meiosis 2. If no, then entire oogenesis will not completed. They will stop it, and this secondary oocyte will be released out from the female body lah. So every month you're doing menstruation mechanism. Every month, human release one secondary oocyte. If there are no fertilization, meaning that this secondary oocyte will be released out and be denatured from the female body. But if there are fertilization, meaning that it will complete the meiosis 2 and produce the second polar body. Second polar body, and this second polar body will be denatured and they will form fertilized egg at here. And then they are also also have the follicle, meaning that in your egg, your oogonium, all these spermatogenesis, they are all stay in the follicle inside the ovary. So in the beginning process is the follicle, small follicle, growing follicle, and then once they already can be the secondary oocyte can be released out from the ovary, meaning that they already reached the mature follicle stage, or another name is the Graafian follicle. Graafian follicle. So the Graafian follicle will release the secondary oocyte. And once it already released, the follicle will be ruptured and it will form the corpus luteum. And when they have the fertilization process, corpus luteum will maintain. But if there are no fertilization, have menstruation, meaning that your corpus luteum will be degenerated, and the repeated process is coming from here, secondary oocyte. Here. Primary oocyte, prophase 1, complete the meiosis 1, and arrest at the metaphase 2 at here, starting here. So the process is repeat until all the primary oocyte is already used up. Once all the primary oocyte is already used up, meaning that the girl will enter the older stage. There are no menstruation happen anymore for that girl. So normally is sometime 45 years old, 50 years old, like that. So the earlier you reach the puberty stage, the earlier you reach the older stage. So this is oogenesis process for the female. Next, we go through the menstruation. So this is the special for female part. So only the female girl every month will undergo this kind of process. Male does not have this kind of process, but you need to know lah. So normally this topic you also already learned in SPM, the hormonal control inside the menstruation. So there are 5 major hormone hormone that involved in the menstruation. So there are these two hormone I think all of you already know. Follicle stimulating hormone FSH, luteinizing hormone LH. They are both of them are secreted from the anterior pituitary gland. So this is very, very important. You need to mention anterior because in pituitary gland they have anterior and posterior. In STPM, you need to mention specified location. If you not mention specified location, you just write pituitary gland. You need to see the answer scheme. Sometimes the answer scheme accept, but sometimes it is not accept. So it depends on that year answer scheme. So it will be say full, you write anterior. But if you write posterior, then totally wrong. And also another hormone, GnRH, gonadotropin releasing hormone. This one is secreted from hypothalamus. And there are also another 2 hormone, progesterone and estrogen. Mostly they are produced from, secreted from the corpus luteum. Okay, so let us see. So this is the entire process of the menstruation. So we need to know that within the 1 month menstrual cycle, within the 1 month, normally is day 0 to 28 so nearly 1 month 1 month. Menstruation normally is occur at here, right? Menstruation here. Menstruation flow phase. This is the phase during the day 0 until day 7, something like that. Is the phase where the female undergo menstruation. Menstruation every month. You have the blood flow, and human is very painful at that day. So this is the phase for the menstruation flow phase. So there are 4 graphs you need to know. The first graph is the graph of the FSH and LH, pituitary gland of the hormone one. Another one is the ovarian cycle. So during menstruation, day 0 until 28, what is happening in the ovary? So meaning that follicle, what is the happening during the follicle stage? All here. And then also there are also another 2 hormone as estrogen and progesterone. So you can see both Campbell, they use the estradiol. So this estradiol actually is estrogen because this is United States terms. In UK we use estrogen. So in exam you can still use this estradiol, but I recommend you use the estrogen more easy. And also the final one is the uterine cycle. This is what happening in the uterine wall. So you need to see it one by one. Okay, first of all, we see during the day 0 until day 13, in this period before the ovulation, because in SPM, I think all of you already know, day 14 is the day for ovulation day. So before ovulation, what happening at here? Okay, so we will see it. So first of all, during the initial day here, normally you are facing the menstruation, right here, menstruation process here. Okay, so what is happening? So first of all, your hypothalamus will start to release the gonadotropin releasing hormone, GnRH. So this GnRH play the role to stimulate your anterior pituitary gland. So once it stimulate, it will release the hormone of FSH and LH. But in the initial day, FSH is more higher compared to LH one. So at here, LH is not doing nothing one. LH is not functioning. They do not function much one. FSH function more. So FSH is follicle stimulating hormone. So their name is already very general. It's already direct. Follicle stimulating hormone. So the function is to stimulate the follicle to grow. So your follicle, of course, containing the secondary oocyte. So your secondary oocyte was starting to do the nothing to do the meiosis process as mentioned during the oogenesis process. So FSH was starting to secrete the growth of the follicle. So this growing follicle will start to secrete the estrogen. So it starting to secrete the estrogen. So that is why that you see here when the FSH is starting to increase, estrogen starting also increase because growing follicle secrete the estrogen at the initial stage. When the corpus luteum not yet produced, estrogen at the initial stage is secreted from the growing follicle. Follicle. Okay. So this is the first 13-day process that produced in the 13 day. And then at the initial day, normally is in the day 0 until day 7. You can see here, the estrogen level actually is very low. It's very low. So this will inhibit the production of SHH and LH. So that's why that during the initial stage, your FSH and LH is not very high one because of estrogen. The estrogen level is very low. So when the estrogen is secreted by the follicle begin drastically, your level of the FSH and LH starting to increase. The high level of the estrogen also can stimulate the GnRH to causing more GnRH is released. So this is a positive feedback mechanism. So this is what happening during the first 13 days. So after the first 13 day, during the 14th day here, ovulation is already occur. So in the ovulation occur, your estrogen level is already very high at here. So once your estrogen is already high, meaning that it already can stimulate your hypothalamus to secrete more and more FSH and LH. So that's why that you see during the day 13, your FSH and LH is rise steeply, especially LH. Especially LH. So normally at day 14, GnRH secrete more LH than FSH. LH is secrete more in the amount. So high concentration of LH will stimulate the ovulation process. So meaning that it will release the secondary oocyte from the Graafian follicle like this. So this like this. So at the same time, LH will also stimulate the development of the rupture follicle into the corpus luteum. So this is what happening in day 14. After that, what happened is that after ovulation, you will see that estrogen will be secreted by the follicle, and estrogen will stimulate the repair and the growth of the endometrium wall. So this is the initial stage lah. What is the function of the estrogen? Why estrogen need to be very high? So it is to play the role in the initiate the development of the endometrium wall. It need to build up the endometrium wall, because at the beginning, day 0 until day 7, this is a menstruation stage. Your endometrium wall is already break down. So they need to, if in the new cycle, they need to build up another endometrium wall. So who build up it? It is estrogen. So this estrogen play the role in the development of the endometrium wall. So this mean that after ovulation, your progesterone and estrogen starting to increase very high, especially progesterone. So where is this progesterone produced? So it is produced from corpus luteum, and also corpus luteum also will generate estrogen. But after the ovulation, progesterone is higher than estrogen already. Progesterone take over the function more compared to estrogen. So what is the function of the progesterone? So normally the progesterone is to maintain the thickness of the endometrium wall. to make sure that the endometrium wall is in the highly thickness, prevent it to rupture of the endometrium wall. So this is a function of the progesterone. So for now, when there are the fertilization process, because secondary oocyte is already released, when there are fertilization process, meaning that corpus luteum is not degenerating, not generated, estrogen and progesterone was still in very high level, and the endometrium wall is not be ruptured. However, during the menstruation process, meaning that there are no fertilization. So when there are no fertilization process, meaning that the progesterone and estrogen levels starting to decrease. They were starting to decrease. Why they starting to decrease? Because the corpus luteum is degenerate already. Already disappear lah. In general terms, is disappear lah. They already hilang. Want to hilang already. So once they already disappear, degenerate, meaning that progesterone and estrogen already will also decrease. And then they are decreased. What happened is in the endometrium wall, there are no the endometrium wall cannot maintain their thickness, and they were starting to rupture. So they will rupture the endometrium wall and form the menstruation. And this cycle is repeat and repeat again. And then the high level of the estrogen and progesterone can inhibit the hypothalamus to secrete the GnRH, so that there are no FAH and LH be produced at here. At here. So this is a hormonal control for the in the menstruation. So it is very easy. In the first few days, hypothalamus starting to secrete the GnRH first. GnRH, and this GnRH will stimulate your anterior pituitary gland to secrete FSH and LH lah. And this FSH and LH play the role in the stimulate the development of the growing follicle, and this growing follicle will secrete the estrogen. So what this function of estrogen? So it is to stimulate the thickness, stimulate the development of the endometrium wall. And once in the day 14, estrogen is already very high, it will stimulate more amount of the FSH and LH positive feedback mechanism. So especially LH, LH is more than FSH. So this LH will trigger the ovulation process. And trigger ovulation meaning that secondary oocyte is released from the ovary. And then LH will trigger the development of the corpus luteum. Okay, then this corpus luteum starting to secrete high amount of progesterone and also estrogen or progesterone higher than estrogen, and this progesterone is to play the role in maintaining the thickness and development of the endometrium wall. And when there are fertilization process, this corpus luteum is not generating, so progesterone and estrogen level are high, and they will do the implantation process lah. But if there are no fertilization, meaning the corpus luteum is degenerate, so that it will repeat the menstrual flow phase, which is the menstruation, and repeat another cycle again and again every month. So that's all about the menstruation process. Okay. So actually this topic you already learned in SPM lah, just a recap only. Because this is very, very important in examination. Normally in exam, they structure question, especially they will come out this kind of graph, and then you need to explain answer the question. Okay, so this is another diagram that taken from the Solomon book. So similarly, what I talked about lah, yeah. So next we go through the human embryo development. So meaning that how the human embryo is formed, how the baby is the birth occur, how the baby is formed. So this is the overall process. So in overall process, first of all, if you want to form a baby human birth, so these are all the process that you need to learn and in the syllabus. So in SPM, normally you only learn the terms only. You know fertilization occur, implantation occur, gastrulation, organogenesis occur, parturition occur. You only know the terms, but you do not know the mechanism. The whole mechanism you also don't know. Okay, so first of all, you want to form the baby, of course you need to have sexual intercourse. The first initial process, you need to have the sexual intercourse between male and female. And then after that, they will have fertilization process. Okay, once they have the fertilization, they will reach the implantation. So you can see here these three. Formation of the blastocyst for implantation. After that, they will go through gastrulation, and finally they will develop the organ. All the organ will starting to form. This is what we call as organogenesis. And finally is birth. This is a birth stage. So the birth stage in scientific terms we call as parturition. So all of this is in the syllabus, and you need to know all of them. Okay. So first of all, so this is a process during the gastrulation and organogenesis lah. So this one is the oral process. Maybe you can know about this first. Later we will proceed more detail about this. How this one is formed. Okay. First of all is the implantation. So this is just a summary process of the implantation. Implantation meaning that you can see ovary starting to release the secondary oocyte, and then you will go through the fallopian tube, and at here is the place where fertilization occur. If there are sperm swim until here, then fertilization is occur. So it will complete the meiosis 2 process to produce the zygote. And then it will undergo a series of the cell division, which is what we call as cleavage, finally to form the blastocyst. This blastocyst finally it will finally enter into the uterus area, and then it will undergo implantation. So this is an overall process. How the fertilization occur. So we need to know the mechanism of the fertilization. Okay, this one is not in the SPM. Okay. So this is the diagram about the fertilization process happening. Okay. So first of all, you need to know what is happen. So sperm from the vagina is swimming until they reach the fallopian tube, and they see the ovum. B is the secondary oocyte lah. They see the secondary oocyte. But you need to know, there are not only one sperm want to penetrate the secondary oocyte. They have many sperm, but they have the mechanism for the secondary oocyte to allow only one sperm can able to penetrate into it. Other type of the sperm that are surrounding at here, they cannot penetrate. If the mechanism is already lost, meaning that they will have many sperm penetrate into it, and this will cause a genetic kind of problem lah. So they will have something of problem lah. But in normal situation, they only allow one sperm penetrate into the secondary oocyte only. So first of all, if you before the fertilization process, your sperm must undergo the capacitation. So it is the process for the maturation and secretion of the sperm in female reproductive tract. So female reproductive tract at here. Your sperm is swimming from here until here, right? So you undergo a capacitation process at here to make sure your maturation and secretion process of the sperm is occur. And then the sperm will become more motile and able to undergo fertilization. So this is a maturation and development of the sperm lah. Make sure that it have enough energy, can swim until they reach the fallopian tube. So after the capsulation process, the sperm will move through the corona radiata. Okay, so you can see this diagram. Because this diagram, they are not using the human one, they're using the, I think it's the jellyfish one, if I'm not mistaken. But actually it's similar lah. Okay, this one is human. So normally in the secondary oocyte, surrounding here, there are many cells surrounding the secondary oocyte one. Not like this one, because this one is they use jelly. In human dog jelly, there are many cell one. So they need to move through the corona radiata. So this all these cells we call as corona radiata. using the enzyme we call as hyaluronidase enzyme. Okay, so then the sperm will reach the zona pellucida. So zona pellucida is the membrane name of the secondary oocyte lah. They call as zona pellucida. So this zona pellucida will be break down by this sperm. How it break down? It will bind with the ZP3 molecule, which act as the receptor in zona pellucida, which will trigger the ecosome reaction. Okay, so what is ecosome reaction? Ecosomal reaction here, you can see. So you can see here where the sperm until they reach here, then they will bind to the receptor we call as the ZP3 molecule that will trigger the ecosomal reaction. So what is ecosomal reaction? So normally ecosomal reaction is that the sperm will release the hydrolytic enzyme to digest the zona pellucida. So you can imagine that this jelly coat is zona pellucida. Before they digest the zona pellucida, they will bind to the receptor first on the surface of the zona pellucida and trigger the ecosomal reaction. So it will release the enzyme to digest the zona pellucida, so that the sperm nucleus can be able to penetrate into the secondary oocyte. Okay, so after that, when your sperm head starting to penetrate into the cytoplasm of the secondary oocyte, the sodium ion channel was starting to open inside the cell. This diagram they're not coming out, but in human they have the sodium ion channel here in the cytoplasm. Plasma membrane of the cell. So this sodium ion will diffuse into the egg cell, causing the depolarization process. So this depolarization process is actually to prevent the additional sperm enter into the cell again already. So this depolarization process is very important. At the same time also, when the sperm is bind already, it will activate the signal transduction pathway, which will trigger the endoplasmic reticulum. You can see trigger endoplasmic reticulum to release the more calcium ion into the cytoplasm here. This diagram not show lah. Here got endoplasmic reticulum, right? So it will release more calcium ion into the cytoplasm. So the increased level of calcium ion will trigger the cortical granule at here within the cortical granule within the egg fused with the plasma membrane. So it was starting to trigger the cortical granule fused with the plasma membrane of the egg. So once they already fused with the plasma membrane of the egg, they will release the enzyme to the space between the plasma membrane and zona pellucida area. So this one is we call as cortical reaction. So meaning that cortical granule in the egg will fuse with the plasma membrane, and this will click off the sperm binding receptor and cause the fertilization envelope to form. This is act as the slow block to polyspermy. So you can imagine like cortical granule is to separate the zona pellucida and the cytoplasm of the secondary oocyte lah. So that the sperm already cannot penetrate into another sperm cannot penetrate into the secondary oocyte already. So this one is we call as cortical reaction. So in human there are two mechanisms to prevent the another sperm penetrate into the secondary oocyte. One of the process is depolarization process that release into sodium ion into the cytoplasm. Another one is cortical reaction cortical reaction. So the cortical reaction make the zona pellucida away from the egg and harden the zona pellucida. So when the zona pellucida is more harder, meaning that the sperm is not easily to penetrate into it lah. So this one is the fertilization envelope is formed. Once the fertilization envelope is already formed, meaning that other type of the sperm, they already cannot penetrate into this fertilized egg already. So this is the entire process of the fertilization. So, okay. Next, we go through the implantation. Okay, so before implantation, there's actually your fertilized egg was starting to undergo a series of the cleavage, like so it will form more cell, many cell. The size of the cell is not changing, but the quantity of the cell starting to produce is changed. So the cleavage process now is occur when the zygote is mitotic divided across the oviduct. So this single zygote nucleus cannot produce enough mRNA to sustain the cell protein need. So the embryo initially will realize on the maternal mRNA and protein stored during oogenesis. So rapid cell division is to allow to restore the proper ratio of DNA to cytoplasmic volume. So this is a mechanism lah. He say that is actually the embryo stage is not enough have the mRNA to do the gene expression process lah. So during the cleavage stage, G1 and G2 phase are skipped, resulting in no increase in mass and size of the zygote. So G1 phase and G2 phase, I think you already know, right? One is you learn in SPM. Before G1, after G1 phase, you will have DNA replication, and DNA replication after that you have G2 phase, and G2 phase after that you will have mitosis process. So G1 and G2 is skipped. So growth phase, the major goal of growth phase is to increase the size of the cell, mass and size of the cell. But for now, G1 and G2 are already skipped, so that the cell is not increased in size. So that's why you can see here this diagram, all the cell size is similar. The difference is just that the cell number is changed. So you can see here normally it's 4 cell stage, 8 cell, 16, 32, 32, all that, 32, all that, until they will form the bastula here. They will form the bastula. So after the cleavage has produced over 100 cells, this embryo is called as bastula. So normally it is a spherical layer of the cell surrounding a fluid field or yolk fluid cavity lah, something like this lah. Something like this lah. So in mammalian like us, normally this one is recalled as bastosis. Bastula is the general term that can be applied all the animal that have do this kind of the reproduction. But in mammalian like human, normally this stage is called as bastosis lah. So this you can see here. So we know that in bastosis, they containing 3 different structure. The surrounding, the outer surface cell is we call as trophoblast. Outer surface is trophoblast. And then the inner part, you can see here that the inner part is we call as inner cell mass. And you can see there are no cell at here. Here, this part is we call as bastocia. So this is very, 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 very important, because all of this part, trophoblast and inner cell mass, later they will develop into different part of the structure through the gastrulation process. So once they complete the cleavage process along the fallopian tube, when they reach the uterus, they will undergo implantation. The blastocyst starting to mechanism of the implantation. So how the implantation start? So normally, at your blastocyst, at the end of the blastocyst cavity is a group of cell called inner cell mass, and implantation of the embryo is initiated by the trophoblast. Not inner cell mass, implantation is initiated by trophoblast because it is the outer layer, right? So the trophoblast was starting to penetrate the endometrium wall. So the implantation, this is after that your implantation is occur. So the implantation is occur when the trophoblast is initiated into the endometrium wall. You can see here your trophoblast, the outer surface one at the inner, within the that near the inner cell mass, starting to initiate into the endometrium wall. So this trophoblast was starting to release the enzyme that can able to break down the plasma membrane of the endometrial wall so that the blastocyst can be penetrated into it. Because you need to know this is still the cell. So how the blastocyst can directly enter inside the cell directly, masuk ke dalam ni. So they need the trophoblast to secrete the hydrolytic enzyme so that the blastocyst is the whole structure of this can directly embedded into the endometrial wall. So finally, all these blastocysts will directly enter inside the endometrial wall one. So this trophoblast, after that, they will extend the finger-like projection, cause the trophoblast tissue can capture the capillaries of the endometrial wall here. So you need to know this still the cell. Cell require energy. They require basic mechanism one. So one of the basic mechanism must be come from the blood. All the carbon dioxide, oxygen, nutrient, must be coming from blood. So they start this trophoblast, trophoblast cause the fingers projection that directly extend into the maternal blood capillaries to obtain all the nutrient. Okay. At the same time, you can see here, inner cell mass is disappear already. Here, right? Initially, inner cell mass is at here. But for now, you can see, why they label epiblast and hypoblast. So, inner cell mass starting to develop into a flat disc shape with the inner layer we call as epiblast, outer layer we call as hypoblast. So all these terms you need to know very well. Before the implantation happen, this product we call as blastocyst. Blastocyst. After that, trophoblast initiate into the endometrial wall, causing implantation. So trophoblast will form the finger-like projection cell to capture the capillaries of the endometrial wall. After that, the inner cell mass is now not called inner cell mass anymore. It will become the flat disc shape with the inner layer we call as epiblast, outer layer we call as hypoblast. And then, how about this trophoblast? Once they already capture the capillaries, this trophoblast was continue expanding into the endometrium, and to form four new membrane we call as extra embryonic membrane. So there are 4 extra embryonic membrane. So what are they? So the 4 extra embryonic membrane, all of them are called, one of them is including the chorion. Chorion, I think you may heard it. Chorion. Another one is allantoid, and then yolk sac, and finally is the amnion. There are 4. Later we will see it. You need to also know their structure and the function, especially the function. So this trophoblast can form this extra embryonic germ layer. So this is the completion of the implantation. Once they already form epiblast and hypoblast, then implantation is completed. So after implantation, we will have gastrulation. Okay, so what is gastrulation? So normally, gastrulation is the process when the blastocyst become a 3-germ embryo, or we call as gastrula. So the final product is gastrula in the gastrulation. So what the gastrula containing the 3-layer embryo. So you can see here. This still not yet. This one. This is the final product. We call as gastrula. We containing 3 germ layer is ectoderm, mesoderm, and endoderm. Okay. For now, you need to know where is this ectoderm, mesoderm, and endoderm developed from. They develop from where? Because we already know have epiblast and hypoblast, right? Okay. So gastrulation normally in human, embryo is developed almost entirely from epiblast. So epiblast is at the inner part, right? Inner part at here. Inner part. Hypoblast is the outer part. Outer part. So some of the epiblast cell will remain as ectoderm while others flow along the permittive streak to form mesoderm and endoderm. So majority of all these three germ layer are formed from epiblast. So how about hypoblast? Hypoblast will develop yolk sac. From hypoblast, yolk sac. So you can see here this one. So three germs layer are come from epiblast. Majority have come from epiblast. So what is the three germ layer? The epiblast will develop into ectoderm, mesoderm, and endoderm. Okay. How about the trophoblast? As I mentioned before. So the trophoblast, they will form chorion, and also anion. So chorion and anion, I already mentioned, is extra embryonic layer. So this is the terms that you need to know lah. So the cells of the invading trophoblast, epiblast, and adjacent endometrial tissue will contribute into the placenta. So very important. Trophoblast also include the development of the placenta. So the trophoblast, epiblast, and adjacent endometrial wall will contribute to development of placenta. So where is placenta developed from? Trophoblast. One of the component is trophoblast and also epiblast. Epiblast. So very important. You need to know every term's different phases. So epiblast is coming from where? Inner cell mass. Hypoblast also coming from inner cell mass. So what is happening after in the gastrulation? What is happening in the epiblast? Epiblast will develop into these three germs layer: ectoderm, mesoderm, and endoderm. So how about hypoblast? Hypoblast will develop into yolk sac here. And then what is the chorion and anion cavity? All these can be developed from trophoblast. And trophoblast also can develop in forming placenta as well. So all these you need to know lah. Okay. So this is finally that containing the three germ layer we call as gastrula lah. So this three germ layer is very important in the organogenesis process because finally this three germ layer will develop into the organ. Different layer will develop into different type of the organ. So this is the overall process of the gastrulation. So next we go through the organogenesis, which is the formation of the organ. Okay, so this one, please memorize it. You need to memorize what is the ectoderm, mesoderm, and endoderm organ will form. So normally this one is coming out in the objective question. Maybe a majority they were coming out in objective question, and you need to determine maybe they will need to ask you which one is correct, which statement is correct, something like that. So ectoderm, so you need to know the location as well. Ectoderm normally is that the inner part one, the most inside one. Mesoderm is very easy, middle, is at the middle part one here, middle part. And then endoderm is at the outer part one. So ecto is at the inside one, meso middle one, endo is the outside one. So you need to know lah. So what is the ecto? Ecto meaning that it is a very in the inside part, the most inner part. So normally it develop into the nervous system, those sense organ, and also the outer layer of the skin, skin and its associated structure like nails, hair, and also pituitary gland. So this is for ectoderm. For mesoderm, mesoderm is the majority. Most of the organ is coming from mesoderm one, including notochord, skeleton, muscle, circulatory system, reproductive, excretory, inner layer of the skin. This one is the outer layer. Ectoderm is outer layer. Mesoderm is inner layer, and also digestive tube, all that. And for endoderm is the lining of digestive tube, tube like that. So normally endoderm is the epithelial lining lah. You can see here. Including thymus, thyroid gland, all that is from endoderm one. So please, this one you need to remember it. You must remember. Okay, first of all is organogenesis. Okay, for the information, you don't need to know all how these organs are formed, but you need to know the initial organ form. The first organ form for human, for embryo is what? So, of course, your first organ is notochord, bone, and spinal cord. This is very important, right? So that's why that you in the baby stage, you can see the brain of the baby is very big one. Compare very bigger size one. During the pregnancy, you will got see the diagram image, the ultrasound image. You can see the baby brain is very big, right? But the body is small, small in size. Yeah, that's why lah. Because the first organ will form in the embryo state is notochord, brain, and spinal cord. These three are the most important thing in organ that will develop in the early vertebrate embryo. Okay, so this is the process lah. During the 90 days, 20 days, and also 26 days lah, after the fertilization process. Okay. Okay, so what is happening in the first day? You see, in the first day, what is happening? What is happening? Happening at here. So, first of all, your notochord is already starting to form. Form. So, notochord normally is your is the mesoderm. Right, I already say mesoderm the develop into notochord. So notochord normally is one type of the mesoderm cell. Mesoderm cell will go forward along the length of the embryo as a cylinder shape. You can see here is cylinder shape. Cylinder shape. And then the inner part is endoderm, right? Endoderm, and then the upper one is neural plate. Okay, so how this neural plate is formed? So normally your notochord eventually is replaced by vertebral column, although the notochord will remain in the cartilage disc between the vertebra. This one is statement is want to tell you notochord normally will be disappear when the in the mature embryo. So that's why that you are very rare to see here. Human got notochord lah. What is notochord? So that notochord. So notochord basically will disappear lah when in the mature state. So what is the function of the notochord? Later we will see it, the development of the notochord. So notochord will cause the overlying of the ectoderm to become thicker, thick, and differentiate to form the precursor of the central nervous system, the neural plate. So notochord play the role in the formation of the neural plate. It will differentiate to form the neural plate. So this thickening is produced by the elongation of the dorsal ectoderm cell or dorsal ectoderm cell. This cell then assume a wedge shape because of contracting bundle of actin filament at the apical end. Okay, so that's all about the first stage. So the first stage is you need to know notochord is formed and then the neural plate is also formed. Notochord and neural plate is formed. Okay, after that, the second stage is you can see the structure is changing already. Structure is starting to change. So what is happening at here? Okay, so the cells of the neural plate here, you can see the cells of the neural plate were starting to undergo change their shape, cause the central cells of the neural plate move downward to form a dispersion we call as neural groove, like here. So it will change the shape, move downward to form neural groove, like this. So this is we call as neural groove. And neural groove normally is the cells flanking the groove on each side from the neural fold. Here. So you can imagine it's like the folding lah. The neural plate is already folding is occur to form the neural groove lah. So the neural fold and the surrounding the neural groove is we call as neural fold. So this neural fold was starting to develop the neutral neural tube here. So you can see here, neural fold was starting to develop into the neural tube. So it will change their shape first. So the neurophore is continue to change their shape, bring the neurophore close together. There are two neurophores. They are close together, until they meet and fuse to form the neurotube, something like this. Yeah. So neurophore is like this, and then they need to fold again and until they meet to each other to form the neural tube. So in this process, the hollow neural tube comes to lie beneath the surface. So neural tube is at the underlying the surface one. So you can see something like this. So there are one like a house, right? Ini macam bubong rumah. So neural tube is dekat dalam lah. Yeah. So the anterior portion of the neural tube will goes and differentiate into the brain, and rest of the tube will develop into the spinal cord. So this is very important. What is happening after neural tube? So the neural tube later will the anterior position of the neural tube will grow and differentiate into the brain. Other parts of the tube will differentiate into the spinal cord. So brain and spinal cord is already formed from the neural tube. Okay. What happening to the what is happening to this endoderm? Okay. So endoderm. Okay, what is happening towards the ectoderm? Another one. Sorry, ectoderm. Okay, ectoderm. So ectoderm, as I mentioned before, ectoderm is from the nervous system one, and ectoderm is located at the most inner part here. Okay, so what is happening for this ectoderm? For ectoderm, they were starting to develop into the neural crest, like here. This part. This is called as neural crest. Neural crest is coming from ectoderm one. So nervous tissue arrives from the ectoderm from each side of the neural tube at here. So neural tube at here. So you can see here this one we are calling as the neural crest. So the neural crest will migrate downward. You can see here, it will migrate downward to form something like this. From the original position and form the dorsal root ganglia of the spinal nerve and the postganglionic sympathetic neuron. They were starting to form the nervous system, and other neural crest cell will migrate various locations to the embryonic give rise of the sense organ. So the most important thing is that the neural crest is majority important for the nervous system. Okay. So another part we call you can see here, there are somite at here. So where is somite coming from? You can see somite. You can see here somite. So where is somite coming from? So somite is the block of the mesoderm formed on either side of the neural tube at here. Here. So, while the neural tube is already formed from the dorsal ectoderm, the rest of the basic architecture of the body is being rapidly established by the change in the mesoderm. So the sheet of the mesoderm on either side of the developing notochord will separate into a series of the valve region we call as somite. And then the somite. So the somite themselves are transient embryonic structure, and after that, they will differentiate into the skeletal system. So the most important thing is that the somite will develop into the skeletal system, and then skeletal system and also muscular system. After that, you also will form the kidney, reproductive structure, and also circulatory organ. So you can imagine that somite actually is formed from the mesoderm one. So different part of the somite, they play important role for the development of different organ. Yeah. So that's all about the development of the organ. So the most important thing is that the notochord, brain, and spinal cord are the first organ developed in the human. So this one is the mesoderm. You can see here. For the mesoderm, you know that it produce many organ, right? So this one they already classified. This one is the mesoderm. So there are 3 part. You can see here 3 part. So you can see here this one is the neural tube, right? Neural this one. Ini. So you can see here. See here. So there are differentiate into 4 major layer: chorda mesoderm, intermediate mesoderm, lateral mesoderm, peristeal mesoderm. So different part they will form the different organ lah. So for chorda mesoderm is forming the notochord lah. Notochord lah. Here. At here one. So it is located at here, right? Now ini here. So it formed here. And then the intermediate mesoderm form kidney and gonads, and lateral one is forming circulatory system, peristeal forming head and skeletal system, all that. Yeah. Okay, next is about the development of the heart. So the heart is originate through the fusion of pair of blood vessels, and normally your heart is forming also from the here, right? Lateral plate mesoderm. It form the mesoderm one. So when the venous blood enter into the single atrium, pass into the single ventricle, which is located above the atrium, and then is pumped into the embryo. So during the subsequent development, the entrail will undergo a process of torsion that bring it to a position above the ventricle. And parturition from that divide the atrium and the ventricle into right and left chamber. So you can see here. Because the heart is originated through the fusion pair of the blood vessel. So initially there are only 2 blood vessels only. Aorta, the atrium and ventricle, like that. Ultra and vein only. After that, when the venous enter the blood into the single atrium, pass into the single ventricle, something like that, something like that. And after they were starting to separate the left ventricle and left ventricle, right ventricle, left atrium and right atrium lah. Finally to form the heart, heart like human just now we see now. So that's all about the organogenesis. So next we go through the placenta. Okay, so where is the placenta developed? As I mentioned, it is developed for trophoblast. When in the 2nd month of the pregnancy. So actually this is a dish-shaped organ that containing the both embryonic and maternal blood vessel. So you can see this is the diagram. So normally placenta is located at here, and this is the enlargement diagram. So you can see here there are maternal part and the embryo part. So the fetal portion of the placenta, another term is we call as chorion. So chorion is one type of the extra embryonic layer. So placenta containing maternal part, this is a maternal part, this is the embryo part, and between them the blood capillary actually are not connected to each other one. So all the nutrient is will pass through this cavity first. After that, they will enter into the maternal into the embryo blood vessel. So normally blood from the fetus to placenta is through the umbilical arteries. Blood flow from the placenta to fetus is through the umbilical veins. So normally is for the function of the placenta is for the material exchange by diffusion, and also to provide the immune system to protect the mothers, all that, or to protect the baby and prevent the harmful bacteria and toxin to enter into the fetus, and prevent the chemical reaction that occur in mother's part into the harmful factors. And also the blood pressure of the mother is very high. If they are connected to each other, the high blood pressure of the mother part will make the blood vessel of the embryo part burst, because the embryo blood vessel is more thin compared to mother one. So if you directly transfer this kind of pressure to the embryo part, your blood vessel of the embryo will burst. Also prevent the mixture of the blood type, because maybe your mother and the baby blood type is different. So if they are mixed together, meaning that they will have oogenesis happen. So they want to prevent this. And this. Also, placenta also play the role in the hormonal control. So later we will see it how it play the role in hormonal control. Okay, so this is the extra embryonic membrane. There are 4 extra embryonic membrane, as I mentioned during the implantation process, right? So the first, so this is a membrane. So chorial, anion, allantoid, and yolk sac. These 4. Chorial, anion, allantoid, yolk sac. So this so chorial is the very outer layer, which is the fetal part of the placenta here. Here. This is we call as fetal part of placenta. But if other than that, the outermost layer that protect the embryo is we call as chorion. And the inner part is the anion. And allantoid is the tube, not say tube umbilical cord that connected between the embryo and also the mother and this is a yolk sac. So yolk sac. So normally you need to know the function. So normally chorion function is like the placenta one because it developed from the trophoblast and consists of the coronal villi like the finger-like projection for the maximize the gases exchange, nutrient exchange, all that, something like that. So anion, you can see here, anion is enclosed to entirely the embryo. It encloses entirely the embryo and it secretes the amniotic fluid. So you can see here, there are many fluid at the inside the baby, right? You see the ultrasound, you also know that during pregnancy, all of that surrounding the baby embryo is the water one. So this water is we call as the amniotic fluid secreted from the anion. So this normally this one is to serve as the protective cushion to absorb the shock and prevent the amniotic membrane from sticking to the embryo. And then another one is allantoid. So normally allantoid is the form the umbilical blood vessel that play major role to connect the mother's part and also the fetus part for the gases exchange, also nutrient absorption, all that. And then finally is yolk sac. So normally is connected to the embryo of a yolk stalk and develop even in mammalia embryo that have little or no yolk, and it is the first embryonic membrane to be appear and containing the plentiful blood vessel, and also it is the place where the formation of the blood cell for the embryo. So that's all about the 4 type of the extra embryonic membrane. Next, we will go through the role of the hormone during pregnancy. Okay, so not only the hormone is happening in the menstrual cycle, hormone is also very important in pregnancy. Okay, so this is the basic graph for you to know that the hormone play the role in the pregnancy. So there are 3 major hormones estrogen and progesterone. I think I don't need to explain many more The new hormone is we call as human chronic gonadotropin here. So you can see that after the fertilization during the pregnancy, HCG, this one is HCG, short form, is increasing in very high amount. But after that, they suddenly decrease. And during the late stage of the pregnancy, you can see the hormone, the progesterone and estrogen starting to increase in large amount. So you need to know what is the role of HCG. So HCG, normally you need to know their location of secretion. Where is HCG produced? So HCG is secreted from the trophoblast, at the trophoblast or placenta. Another term is not placenta, trophoblast and or chorion. Trophoblast and chorion before the placenta is formed. So this is the HCG hormone where is formed. Okay, so the major function of the HCG is to prevent the degeneration of the corpus luteum. Why you can see that why we say that? Why we say when there are fertilization process, corpus luteum is not degenerated. Why? Because of this hormone. Because if there are no this hormone, corpus luteum will automatically degenerate. They are not care have fertilization or no fertilization. Because the set point is that corpus luteum after the ovulation process, whatever he have fertilization or not fertilization, corpus luteum will automatically degenerate one. So they need to prevent the degeneration of the corpus luteum. So this hormone happen. So how this hormone produce? It only produce when trophoblast and chorion. So where is trophoblast and chorion form? Only when in during gastrulation process, implantation process. When they have implantation gastrulation process, then trophoblast and chorion are formed. If no that process, then no trophoblast, no chorion, then no HCG lah. So this HCG hormone is play the role like this. So since the corpus luteum is not degenerated, so they can still maintain the high level of the progesterone and estrogen to maintain the thickness of the endometrial wall during the early of the pregnancy. So you need to know that during the early pregnancy, the role of the function of the progesterone and estrogen mainly is to maintain the thickness only. But when during the late of the pregnancy, during the birth, you can see here progesterone and estrogen starting to increase. They have different function. So that function later I will proceed to it. So this is the HCG hormone function. But after 2 months, after the first 2 months, so normally you can see here week 12 like that, it was starting to decrease. So once the HCG hormone decrease, meaning corpus luteum also degenerate. Okay. When corpus luteum degenerate, so how the progesterone and estrogen produce? Where is progesterone and estrogen produced? Because corpus luteum already gone. They're already gone. Corpus luteum already cannot produce progesterone and estrogen anymore because they're already gone. So where the progesterone and estrogen still can be secreted in high amount. So the answer is placenta. So after 2 months, when the placenta is already formed, so one of the function of the placenta is to secrete the progesterone and estrogen to replace the function of the corpus luteum. So now, once the placenta is already formed, then corpus luteum can be degenerate. Degenerate. Why it degenerate? Because of HCG hormone. So the HCG hormone reduced already. So they have the related lah. So the major function of this hormone is to prevent the degeneration of the corpus luteum only. The major function lah. Also they have another side function including the entry of the amino acid, development of the mammary gland, and also stimulate the glucose and lipid metabolism lah. As well. So this is a other side of the function of the HCG lah. But the major function is to prevent the degeneration of the corpus luteum. Okay. So there are total 6 hormones involved in the pregnancy. So I already mentioned HCG. So of course, progesterone and estrogen. Another 2 is oxytocin and ACTH, which is a factor adrenocorticotropic hormone. So yeah, HCG I already mentioned. So I think I don't need mention more. So of course, estrogen. So what is the function of estrogen? Okay. So normally during the early pregnancy, the major function is to maintain the thickness of the endometrial wall and also prevent the release estrogen and progesterone also can prevent the secretion of FSH and LH. Like just now I already mentioned in menstruation part. To prevent the menstruation and ovulation to occur during pregnancy. Pregnancy process. So also function with the progesterone. It also can promote the growth and development of the mammary gland. But the role function of the mammary gland is to secrete milk. But during early pregnancy, cannot secrete the milk. You can you imagine that? Have you see before that during pregnancy they have milk produced by the mother one? No, normally milk is produced after pregnancy because that milk is for the baby nutrient one. So during pregnancy they have no milk, but they already have mammary gland. So want to prevent the mammary gland to secrete the milk, one of the major function is the for the progesterone and estrogen is to prevent the milk to secrete the mammary gland. So they develop mammary gland but stop the mammary gland to secrete the milk. So their function lah. Also, they also for estrogen, they also will stimulate the enzyme production that break down the collagen fiber in the cervix wall to make the cervix more flexible. So this is the um to prevent the baby coming out lah. Okay. Okay. For progesterone, earlier pregnancy is similar something like estrogen one. Maintain the thickness. Just how much I say, inhibit the release of LH and LH. Also, development of mammary gland. Also, prevent the milk secretion. But there are also other type of function, including the oppose the stimulatory effect of estrogen on uterine smooth muscle until late in pregnancy. And stimulate the secretion of prolactin from the anterior pituitary gland. Okay, so there are also another hormone we call as prolactin. So normally this prolactin is play major role from other part that actually you don't need to know lah. Not say that you don't need to know. Play the major in lactation. This one is what it say that is the to produce the milk from the breast. Produce the milk from the breast for the baby to eat, to drink the milk. So prolactin later you will see it during the lactation process. We will also include it in the syllabus. So the function of the progesterone actually is to secrete the prolactin lah. Prolactin, and also promote the growth of the mammary gland, inhibit the contraction of muscle tissue in the uterus and oviduct. Because at the early pregnancy, you are still not birth there. So progesterone can be function to inhibit the contraction of the muscle cell in the uterus and also increase the production of the mucus in cervix to form a plug that protect against infection. Yeah, so this is a function of pregnant during early pregnancy. But after late of the pregnancy, you can see that estrogen is higher than progesterone already, and progesterone starting to decrease. So estrogen play more function during the pregnancy, especially during the birth. So level of estrogen is the highest point during the parturition here. Parturition is the birth lah. The baby starting to birth, coming out from the mother's body. So this hormone estrogen will play the role with the oxytocin here. You can see here. It will play the role with the oxytocin to provide the positive feedback in the labor stage, which is the parturition process. Okay. So in response to suckle by the newborn, your estrogen level is very high, will cause the hypothalamus to secrete the hormone called as prolactin hormone. To signal to secrete the prolactin hormone. So you need to know that it is not something like the in progesterone, because in progesterone it also already secrete the prolactin, right? So estrogen also play the role in secretion of prolactin at the late stage. Also, it will stimulate the uterine muscle fiber to produce the protein to form the gap junction between the cell to allow the uterine muscle contract together in a coordinated way during the birth, produce highly coordinated contraction. The finally is function is to stimulate the uterine move muscle for parturition by promoting the production of oxytocin receptor. So it activates the oxytocin receptor on the uterus. So there are three major functions during the late stage of the pregnancy for the estrogen. So for oxytocin, so what is the role of the oxytocin? So normally, oxytocin play the role during parturition process. Okay, so you can imagine that during the birth stage, masa nak lahirkan kanak-kanak, kan sangat sangat susah kan. The mothers is very difficult and very painful because you need to see the baby is large like this, but the hole, the coming out pathway is very small, so they need to open up this hole, the vagina very big one. So one, they also need to push. Right, during masa lahir, ibu kena tolak kan, dia kena tolak baby tu keluar daripada um coming out from the mother's body. So how they coming out? So you need to contract the uterus muscle. So contraction of the uterus muscle is very important. And also require high contraction. The contraction period is very long and the stimulus must be very high. So how to make the contraction of the uterus muscle can be for a longer period and the stimulus is very high. Oxytocin play the major role, and this is one of the major example that normally all among the STPM use in the examination. It is a positive feedback mechanism. One of the famous example. So when during the parturition process, oxytocin starting to secret. So where is oxytocin secret? So normally oxytocin is secreted from posterior pituitary gland. From the posterior pituitary gland of the mother and fetus, both of them. But majority is mother part. So when the parturition process is happen, your fetus starting to push against the cervix. Cervix is at here. They starting to push against the cervix, which stimulate the stress receptor in the cervix. So your stress receptor will trigger the posterior pituitary gland to secrete the oxytocin, and oxytocin major function is to stimulate uterus to contract. Contraction of the myometrium wall, and also the increase the power of the contraction, and also they will stimulate the placenta to produce prostaglandin, and this prostaglandin also will stimulate the contraction of the wall. And when the uterus is the contraction of the uterus will provide the positive feedback mechanism to the posterior pituitary gland to secrete more and more oxytocin. So similar to here. Because it stimulates the uterus to contract and causing the oxytocin to secrete more. So this is the positive feedback mechanism. Trigger more. So uterine contraction provide the positive feedback mechanism, which will further trigger more oxytocin secretion and stimulate the further uterine contraction until the uterus expel the fetus. Once the fetus is already coming out, baby coming out, then oxytocin stop. But if not, if the contraction still there, then it will starting to contract, release more and more oxytocin, until the baby is already totally coming out, the parturition stage is ending, then we will stop. Oxytocin is stopped to secrete. Okay, then there are another hormone. This one is the ACTH. So normally this one is secreted by anterior pituitary gland before birth. So stimulate the development of the adrenal cortex, increase the cortisol secretion for the fetal lung maturation. Also stimulate the fetal adrenal gland to secrete the corticosteroid into the maternal circulation, decrease the level of progesterone, and resulting the increasing secretion of prostaglandin. And prostaglandin will contract the uterine wall. So this is just a function of the ACTH. So next we go through the parturition process. Okay, so in parturition process, there are three major stages, which are dilation, expulsion, and placenta. Okay, so what is happening in dilation stage? Okay, so you can see in dilation stage, typically it lasts for 8 hour to 10 hour. So you can see here, this is cervix, right? This is the uterus. So dilation stage is the preparation stage for mother. Normally at here, the mother is already starting to feel, or the baby is already want to coming out already. So what happening is that the contraction starting to happen. The uterus wall muscle contraction was starting to move the factors toward the cervix. Cause the cervix to dilate. So the cervix will become more effective, and it will thin out so that the fetus head can be passed through. So the anion starting to capture, like the one of the embryonic membrane, the anion starting to rupture, meaning the embryonic cavity is already released out. So that's why that during earlier of the pregnancy, when the embryonic cavity is already coming out, meaning that there is the signal the baby already want me to coming out. So this is a signal of the parturition process. So as the Pacific and at this stage, oxytocin starting to produce. So as the baby head push against the cervix, a reflex action cause the uterus to contract. And then the contraction force the head against the cervix again, resulting in another contraction causing the positive feedback mechanism. So something like this. When the contraction is already occur, it will secrete more and more oxytocin and stimulate more and more uterus contraction. So yeah, so this is the initial stage of the dilation. So happening in dilation. So normally dilation is a preparation state. Uterine wall starting to contract. The baby is already reach the cervix. Cause the cervix to dilation of the cervix. So that's why it say dilation. Okay, next is expulsion. Okay, this phase is where baby already coming out. The birth process. Okay, this stage is very fast. Normally it's only 1 hour like that. This stage is preparation stage can last for 8 hours. The most painful stage is at here. Because for now, uterus is already starting to contract. They want to contract it so that they have enough power to push the baby out. Once they already have enough power, then it will reach the expulsion stage. But for this stage, the contraction of uterus will cause painful lah. So expulsion stage is where only 1 hour, but it is the most painful period. This one is not very pain yet, but the period is very long. You need to pain a long time. But for here, it is very pain, but it is very short because the baby is already want to come out. So at here, the contraction of the uterus was starting move the factors toward the cervix and causing the maximum intensity. So it is already maximum, meaning that already ready. Uterus muscle is already ready to push the fetus out. Okay, the mother is already ready. So the fetus will pass through the cervix and vagina, and the baby is born. So this stage is the baby is already born. So with each uterine contraction, the woman bear down, so that the fetus is expelled by the combined force of the uterine contraction and your contraction of the abdominal wall muscle here. Outer part one. Not only uterine, your outer part muscles also play the role in pushing out the baby. However, for now, your baby is still connected to the placenta by the umbilical cord. So the contraction of the uterus sequence much of the fetal blood from the placenta into the infant. So you need to know you still have placenta. And we know that placenta is rich in blood because there are many blood capillaries here. Because placenta plays the role in exchanging the oxygen, carbon dioxide, nutrient all at there. So they're containing mother blood vessel and also fetus blood vessel. So when you push the baby out, of course placenta is still pushing because umbilical cord still attached to the placenta. You still push the placenta out. So that's why that you will see during the expulsion stage, they have many blood at there. So squeeze much of the fetal blood from the placenta into the infant. So the cord is tight and cut, separating the children from the mother. So after when the baby is already coming out, they will cut the umbilical cord. So finally, they will reach the placenta stage. So placenta stage is to remove the placenta from the mother part. Because placenta cannot stay at the uterus one. They need to remove. So they need to placenta starting to loose and detach from the uterus wall and another series of the contraction of the uterus is occur to remove the placenta. So this stage normally some books say after birth because the baby is already come out. You just want to remove the placenta and umbilical cord only. And once all the placenta and umbilical cord already removed, then parturition process is completed. The baby is formed already. So that's all about the parturition, 3 parturition stage. Okay, next is about lactation. Okay, so this one is where after the birth, right? After the birth, we know that after the birth, mother's breast can produce the milk, right? We know all of us know, and all of us majority of our cat will drink the milk from the breast, right? During the baby stage. So this process is we call as lactation. Okay, so you can see this is the breast structure lah. Breast structure. So normally each breast consists of 15 to 20 lobes of the glandular tissue. Consists of many hollow and spherical glandular structure called alveolus connected to the milk ducts. So you can see here, in one breast there are many lobes of glandular tissue. And in each glandular tissue, they have many structure we call as alveolus. This alveolus is not the lung alveolus. Because why we call alveolus? Because it look like lung alveolus. But this one is not lung alveolus. This is the breast alveolus. Okay. So the most interesting thing is that we know that during pregnancy, a high level of progesterone will stimulate the development of mammary alveolus, mammary gland, right? You know, this one is we already know. Mammary gland here. Mammary gland. They will develop the mammary gland during the high level of progesterone. A high level of the estrogen will stimulate the development of the alveolus duct here. This one is I already know. I already mentioned in the function of the estrogen and progesterone during the pregnancy. So, however, the estrogen will block the action of the prolactin on the mammary gland, so it inhibits the prolactin secretion by promoting the release of the prolactin inhibiting hormone from the hypothalamus. Okay, so you can see here. This is the hormonal control of the lactation process. Okay, so what is happening here is that when during the birth, sensory impulse associated with the baby sucking trigger the posterior pituitary gland to release oxytocin and prolactin. Okay, so after the birth, when your baby is starting to suck the mother's breast, it was starting to release the oxytocin and prolactin here. So why they release oxytocin and prolactin? So the oxytocin plays a role in stimulating the myoepithelial cell surrounding the alveolus to contract, thus causing the milk to be ejected by the breast. So you need to know oxytocin play the role for the myoepithelial cell to contract, to eject the milk. To eject the milk. So another one is that what is the function of the prolactin? Okay, so prolactin will stimulate the alveolus epithelial cell to secrete milk into the alveolus lumen. So easily, prolactin is for hormone for to produce the milk by the alveolar cell. Oxytocin play the role in ejecting, eject the milk. Prolactin produce the milk. Oxytocin eject the milk. That's all. So it is very important. So this is a breastfeeding of the baby to the human by the hormonal control we call as lactation. So the first milk produced after birth normally is a yellowish milk yellowish color called colostrum, which is very nutrient rich, rich in the maternal antibodies. So that's all about the lactation. So the most important thing is that when they have the milk sucking process, prolactin and oxytocin are secreted because they have the signal. Your central nervous system will receive the signal or to stimulate the secretion of the prolactin and oxytocin. So that's all about the lactation. Breast function as the lactation. Next, we will go through the second big topic, which is the sexual reproduction in flowering plant. Okay, so before we go through the reproduction, we must know the flower structure lah. So this is a general flower structure. I think you already learned in SPM. Yeah, right. So we know that in flower, they are also have male and female. So the male organ is stamen, we call as stamen. And for the female organ, we call as carpel. Right. So for stamen, they're including the anther and filament. And for the female one, they're including stigma, style, ovary, and also ovule. So this is the major role of the stamen and carpel, so you need to know lah. So of course, stamen, we know that for the male one, female one, the reproductive organ that produce in flower is that pollen gland, right? Pollen gland. For the carpel one is ovary at here, the embryo sac. So we know that in flower, they are containing the male reproductive organ and also female reproductive organ called stamen and carpel lah. So we need to know about the life cycle of a plant. So this topic, we will learn more detail after in semester 3 lah. Because in semester 3, they will have one of the chapter 1, the chapter 1 of the semester 3, you will learn about more about this. But for this, I just give you to the basic information only lah. So they will have the plant life cycle is we call as alternation of generation life cycle, meaning that in plants, they will spend part of their life in a multicellular haploid stage, and some of their life is at multicellular diploid stage, meaning that in some of their stage is called haploid, some of their is diploid, meaning that haploid is gametocyte, right? Haploid stage and they only have half of the chromosome number. Diploid stage is that 2n, meaning that they have full number of chromosome number. So diploid stage is in the adult stage, which we could call as sporophyte, and they have another stage which we call as gametophyte. So we know that in plants, in male and female sexual organ, they containing spore, right? For male, we call the spore is called as microspore. For here, female, we call as megaspore. Right? This one, all of you already know that. Female is called megaspore. Male is called as microspore. Right? Microspore. So you can see here, during the gametophyte that has spore, this spore was starting to produce the gamete. So the gamete in flowering plant, for male one is pollen. For female one is megaspore. For the male one is not microspore. For the male one is stamen here, and then for the female one is carpel. couple. So after that, they will produce the sperm and egg, do the double fertilization process. Once they complete the double fertilization process, they will form the sporocyte. And then after that, they will undergo the spore meiosis again to form the spore, and then repeat and repeat. So they have the one of the stage is called as gametophyte. Along this stage, they are all in haploid number. Along this stage, they are in diploid number. So you can see this diagram. Yeah, this diagram is more easy. More easy. You can see here. See here in the beginning of the process. They are forming the they are forming the gamete, right? So for the female one, the gamete is megaspore, right? Megaspore. For the male one, gamete is pollen gland. So how this megaspore and pollen gland are formed? They are formed from the megasporangium. Megasporangium, and from male is from the microsporangia, which is we call as pollen sac. So how does the pollen sac and the pollen gland formation and the embryo sac formation? They are forming from the microsporocyte and also megasporocyte. So all this you need to know very detailed. Okay, so first of all, as I mentioned, all the plant they also have sperm, they also have ovum. So in humans, sperm is formed through spermatogenesis, right? But in plant, they are not formed through spermatogenesis. They are formed pollen gland. So where are the pollen gland formed? They are formed from pollen sac here. So pollen sac here. So the pollen sac, the another name of pollen sac, they can call as microsporangia. This one is microsporangia. Microsporangia. So when they are microsporangia, so this microsporangia will produce many containing many microsporocytes. They contain many microsporocyte cells. Microsporangia is the general terms of pollen sac. Pollen sac is specific term for the flowering plant, because you need to know plant not only have flowering plant, they also have non-flowering plant. They also have another type of plant. Later you will learn in semester 3. But for now, you just focus on flowering plant. So in flowering plant, the gametophyte of the flowering not gametophyte, the spore of the flowering plant for male, in general terms, we call as microsporangia. In specific term is pollen sac. So in pollen sac and in microsporangia, in one sporangia, there are many microsporocytes. Microsporocyte is cell's name. It is a cell's name. The cells of the microsporangia. Okay, so this microsporocyte will undergo the meiosis process. Okay, after that, this process is we call as gametophyte stage. Here still haploid stage, not haploid, diploid stage, which is a sporocyte stage. Once they already undergo meiosis process, here is the we call as the gametophyte stage. So they will undergo meiosis to form the microspore, and then after that, each of the single spore will undergo mitosis to form the pollen gland. So we know that in major pollen gland containing two different nucleus, generative nucleus and tube nucleus. So of course there are two cells, generative cell with tube cell. So this is the formation of the pollen gland. So next, we will go through the embryo sac formation, which is the female part. So for female, they are similar, so same as male part. They also have spore. So the spore name for female one is we call as megasporangium. For if scientific terms for flowering part is young ovule. Ovule. Okay, this ovule containing one megasporocyte. They all containing one megasporocyte, and this megasporocyte will undergo meiosis to produce the megaspore. Okay, so in meiosis process, we know that meiosis 1 and meiosis 2 all together will form 4 daughter cells. But there are only one megaspore is function. The another 3 megaspore will be degenerate. Okay, so this megaspore after that will undergo 3 times division to produce 7 cells with 8 nucleus. You can see 1, 2, 3, 4, 5, 6, and 7. But in the middle cell, central cell, have 2 nucleus we call as polar nucleus. And then the upper one, the 3 cell upper one, is we call as antipolar cells. Antipolar cells. And then the lower part have 3 cells, right? The bottom one is we call as 8 cell. Another 2 is we call as synergic cell. So this is the structure of the embryo sac. So the female gametophyte for the flowering plant is we call as embryo sac. So for male is we call as pollen grain. So this is the structure of the flower. Between how the pollen grain and embryo sac are formed. After that, this is the embryo sac structure, more larger. After that, we will go through the fertilization process. Okay, so in plant, we call flowering plant, we call as double fertilization. Okay, why we say double? Because fertilization is happening twice in plants. Okay, so first of all, when the pollen grain is already transmitted from the female reproductive organ couple go adhere to the stigma. This one is we call as pollination. When the pollination is occur, okay, so what happening is that pollen grain was starting to absorb water in the stigma and stimulate the pollen grain to germinate by producing the pollen tube. So this pollen grain is starting to produce the pollen tube so that the pollen grain can be passed through to the egg cell, to the embryo cell. Okay, so the pollen tube will elongate along the style and go toward the micropill by chemical attraction produced by synergics. So the arrival of the pollen tube will cause one of the synergic cell die. So when the pollen tube already reached the micropill, one of the synergic cell will die lah. So it provide the line, the way for the pollen grain can be passed through. So as we know that pollen grain containing 2 nucleus, tube nucleus and generative nucleus, right? So what is the function of these 2 nucleus? So first of all, the generative nucleus were divided by mitosis to form the sperm. They will form 2 sperm. So another nucleus is we call as tube nucleus here. Tube nucleus. What is happening here? The tube nucleus will lead ahead the 2 sperm, move along the pollen tube, reach the micropill. And this tube nucleus will penetrate the micropill. You need to know here actually got something covered at there. So the tube nucleus need to like a shoulder to open the root for the sperm cell, so that this tube nucleus will penetrate through the micropill to allow these 2 sperm cells can enter into the embryo cell. So we know that they have 2 sperm cells. So one of the sperm cell will diffuse with the egg cell to form zygote. And another nucleus will penetrate to the central cell with the 2 polar nucleus, which form the triploid nucleus, which is endosperm. So this is why we call as double fertilization, because fertilization happening twice at here. First sperm cell will diffuse into the zygote. Diffuse into the egg cell to form zygote to end. Another one is diffused into the central cell that containing 2 polar nucleus, forming triploid nucleus cell we call as endosperm. 3N. So there are zygote and endosperm. Okay. So this is a way of double fertilization. Okay. Next is the process we call as seed embryonic development. Okay. How the seed is formed. After the fertilization, then you need to develop the seed, right? So how the seed is formed. Okay, so this is the overall process, how the seed is formed. Okay, is formed. So mostly the formation of the seed is through zygote. Endosperm not play the role, the 3N1 not play the role in development of the seed. Endosperm play the role in providing the nutrient to the zygote during the embryonic development. Okay, so you can see here where the zygote once the zygote after the fertilization, endosperm zygote starting undergo mitosis to form the multicellular embryo. So the first mitosis of the zygote will form the basal cell and terminal cell. Here you can see here, basal cell and terminal cell. Okay, after that the basal cell, this one, the lower one, basal cell will divided to the suspensor cell. Here it will divided to suspensor cell, which anchor the embryo to the integument of parent plant. So you can say that and suspensor cell is the like the stem of the embryo, something like that. At the same time, your terminal cell will also divided mitotically to become the pro-embryo that attach to the suspensor cell. And then your suspensor cell will transfer nutrient from parent plant or endosperm to the embryo. So most in particular is embryo. Suspensor cell just anchor the embryo cell only. So it will push the embryo into the embryo sac cavity. Okay. After that, this embryo cell will undergo mitosis to form the globular stage embryo. So you can you see here lah, more easy. Boy and boy, it will form the globular stage, which more larger. Not here there. This one is heart shape. It will form more larger of the embryo cell. So this stage is we call as globular stage. So the globular embryo will develop through mitosis into one cotyledon for monocot. If for dicot, it will form two cotyledons. Okay. So the cell will begin develop into the specialized tissue during this stage. Okay. After that, it will still develop into the heart stage. The embryo will develop into the heart shape cell. We call as heart shape embryo, heart stage. After that, it will develop again through the tapeworm stage. So it will continue to grow as the cotyledon elongate. So cotyledon elongate. So as the embryo enlarge, it often curve backward itself and crush the suspensor cell. So the embryo cell between the cotyledon will differentiate into apical meristem of the shoot. Embryo cell will near the suspensor will develop into apical meristem of root. So for now, in the globular stage, they already form the cotyledon. They're starting to form the cotyledon. So for the monocot plant, only one cotyledon form. For the dicot plant, they will form two cotyledon. So in the torpedo stage, at the end of the torpedo stage, the embryo cell that between the cotyledon here will differentiate into the apical meristematic of the shoot. But for here, it will differentiate into the root, apical meristematic cell of the root. So it like that. So finally, it will form the cotyledon. Cotyledon some like this. Okay. So this is the final stage, the mature embryo. So you see here. So there are two cotyledon for the dicot, and then this is a stem apical meristem style, and then this one is root one. So between two cotyledon is the stem apical meristem, and then at the end of the suspensor cell is root apical meristem. So the outer integument will form the testa. The inner integument will form the tegment. So it's here, outer part here. Here it form the testa, and the inner one is from the tegment. And then the central part of the ovule is broken down to be used by the embryo. and then the ovary wall develop into the pericarp, and then finally the seed tissue will undergoes hydration and seed dormancy is reduced, and finally we will form the seed like this this. So this is the overall process of the embryonic development. And then next is the fruit formation. How the fruit is formed. So normally we know that fruit is formed through the ovary. So after the double fertilization, within the ovule will develop into a seed. So that's I say before lah, embryonic development ini lah. Semua ni. After that, and the ovary or surrounding will develop into a fruit. So normally fruits consist of three parts, endocarp, mesocarp, and endocarp. Exocarp, the outer one, mesocarp, the middle part, and then the endocarp is the inner part one. So normally epicarp is developed from the outer epidermis of the ovary wall to protect the fruit lah. Mesocarp is the middle tissue of the ovary wall, and the endocarp is the inner epidermis of the ovary wall lah. And fruit development continues until the seeds mature. Okay, next is the new topic that never never saw it before in the old syllabus. So this is totally new topic, plant tissue culture. Okay, so what you will learn in this topic is that based on the MPN syllabus is that you need to know the basic principle of the plant tissue culture. Let me see. Okay, describe the tissue culture. Of course, basic principle lah. What is plant tissue culture? And grafting in asexual reproduction. Okay, so what is plant tissue culture? Plant tissue culture. So you can imagine is that it is the synthetic human-made plants. In more faster way. Meaning that normally if you think that you imagine if you want to make a plant, you want to go up one plant, normally you require what? You require a land, especially if the plant is the very big plant. You require a land, you require soil, you require many things. This is very many things. So in scientific study, if you want to planting a plant like this, wow, very difficult because it takes time. Not only complicated process, but it takes time. You need to function like you are a gardener. You are the florist. You need to take many times to take care of your plants. So in scientific study, if you want to speed up the research of the plant, we require plant tissue culture. So in real world, we also have animal tissue culture one. Yeah. But in your syllabus, you are not learning animal tissue culture. but and add on plant tissue culture. Plant tissue culture. Okay. Okay. So what is plant tissue culture? So in is very simple way to think it, you can take any part of the plant, not necessarily seeds. Because in normal people, we know that if you want to planting a plant, you require seed of the plant, right? You require seed of the plant, then you can grow up the plant. But in plant tissue culture is that you don't need to use the seed. Any part of the plant can grow it. You can use any part of the plant, either leaf, either their flower, either you use the shoot, either you use the root of the plant, you use the stem of the plant, you grow it in the environmental control supportive condition, which we call as in vitro condition. You still remember some of you people know that got in vitro and in vivo. In vitro is like this, meaning that you grow your plant in the environmentally controlled condition like this. So you go in the plant in the lab only. You don't need go to outside. You don't need require soil. You just grow the plant in the lab. You just prepare the medium, prepare the nutrient that required by the plant, and you prepare any hormone. That's why in last chapter, say before auxin and cytokinin, both of them are play the role in tissue culture one. Very important because auxin, when your auxin is higher than cytokinin, it provide rooting. But if cytokinin more than auxin, it provides shooting. You still remember. So this is the overall process, meaning that you have plant. After that, after the plant, after that you take any part of the plant, any plant, maybe leaf, maybe shoot, maybe flower, maybe stem, any part, after part, and then it will form the form the catalyst, and then it will form the organ, directly form the organ one. Or you can go through the liquid media, suspension culture, all that lah. So this one is more clear lah, more clear picture for you guys. Right. So you can imagine is something how to start here, right here. Okay, you take any part of plant. So for this diagram they use the leaf. After that they will induce the de-differentiation process, which is the catalyst. They induce the catalyst. So what is catalyst? Okay, catalyst normally is the intermediate between intermediate structure of the plant. What is the intermediate? You can say, is it a seed? Is it a shoot? Is this a root? No, there are no. So normally it is the unorganized, undifferentiated cells one. So you can, meaning that in general terms, what is the catalyst? It is the undifferentiated cells of plant. So meaning that it can differentiate to any way based on what we want. So if you want to produce the leaf, if you want only leaf, then we can modify something, modify problem, then you can produce only leaf. But if you want to store the catalyst, yes, we can store it. But if you want to produce the whole plant, also can. They can still produce a whole plant. So in plant tissue culture, you can see, you can imagine that it is the scientist, the way scientists play around the chemical nutrient of the plant. You can adjust any chemical nutrient that you want, or hormone that you want, to achieve the desired objective. So you can see, responding to the different stage of the plant, leading to plant regeneration for exclusive leaf management, regeneration. Okay. So in simple word, this is the technique that the plant do not require any reproduction, all that mentioned before, double fertilization, embryonic development to produce a plant. No need. Just require only the small pieces of leaf. Then you can produce this. You can directly produce a new plant. Just small pieces of leaf only. So they have many technique lah. So this is the overall basic principle of the plant tissue culture. Tissue culture. Okay, so there are major, these types of the tissue culture lah. Tissue culture. So one of the type is embryogenesis and organogenesis. Okay, so embryogenesis and what are the meaning different between these embryogenesis and organogenesis? Okay, embryogenesis, of course, want to develop embryo, meaning that you need to develop the embryo, which the cells is already developed into differentiated plant, either sexual or asexually, to form a new plant. New plant. For organogenesis, you just want to form organ. So organ of the plant normally just only two. Either you want to form shoot or you want to form root. You want to form shoot or you want to form root. Or you need to form flower, or you want to form leaf only, just only leaf. But for embryo, is a new plant, totally new plant at all. And this one is we call as angiogenesis. Okay, so angiogenesis is another term that we use the haploid gamete, which is either for female. For male one is we use the pollen gland. For the female one is we use enter. So there are enter. Okay, sorry. Okay, so there are three major types of the tissue culture that I taken from example lah. Actually, there are many types and many types, but these three types is the majority that we use in scientific research. So first one is the somatic one, not somatic, embryogenesis. Second one, organogenesis, and then the final one is androgenesis. Okay, so embryogenesis is the development of embryo, meaning that you need to develop a whole plant, including the leaf, including the shoot, all that. But for organogenesis, you just either want to develop the organ only, either root, either shoot, either leaf, or either only flower, only one only. You don't want the maybe you need to do a research about the root. So I don't want the shoot one. I don't want other part of plant. I only want the root only. So this one is called organogenesis. Embryogenesis is whole plant, the whole development of the plant. Okay, androgenesis, another one is androgenesis. This one is to development of haploid plant originating from the pollen gland through a series of cell division and also the differentiation. Either using pollen gland or using the anther. You directly, meaning that you directly use the pollen gland, you directly extract the pollen gland, or you use the anther, the male sexual organ, to produce the haploid plant. Meaning that this chromosome plant, although similar, but the chromosome number is double of the haploid, meaning that you something add lah, add something like that. So this is a type of the tissue culture. So you see here for somatic embryogenesis, they have direct or indirect. So the meaning of direct indirect is that direct is directly you use the plant part. Either you use leaf, either you use shoot, you use stem, you use root, or you use flower to develop the plant. Indirect method is you use the catalyst. So catalyst in this plant tissue culture is very amazing part because it induces de-differentiation. So this is an undifferentiated cell. So it can generate any type. You can generate shoot. You can directly generate shoot, or you can shoot. This is the organogenesis, or you can use embryogenesis. Use embryogenesis to induce the embryo, somatic embryo. So directly you can produce a plant, or even you induce the shoot, you can still develop the root and to form the plant. So plant. So something like that. So you can change or so direct method means you directly use the plant, directly go through meaning this regenerate directly use the plant part and directly to form the shoot. But for indirect method, meaning you use the catalyst. You form the catalyst first and then you store it there. Okay, after that, after few months, after few years, or you want to do a research, then you use it. Then you use this catalyst, you can directly generate different type of the plant. Okay, so this is the major type of the plant tissue culture lah. Okay, and then this is the advantages of the plant tissue culture. So normally, one of the advantages is you can do produce the particularly good flower, fruits, and have other desirable trait easily lah. You can exact copy. Because this one is directly copy one. There are no variation process. Because you use the normal reproduction process, you can have a slightly modification change right between the mothers and children. But for this, if you successful, the daughter cell is totally same like the mother cell one. You can say it is like a cloning technique. Like the cloning technique lah. Quickly produce major plant. Yeah, it is very fast. You save the time, and also absence of seed. You can produce the plant, genetically modify it to produce the fruit that do not have seeds. Yeah, you can do it using the hormone, right? Penotopy, oxy, cytokinin, all that. And also regeneration of whole plant that have genetically modified. You can take the plant of the cell, right? And then you do the genetically modified genetic engineering of that cell. And then after that, you do the plant tissue culture to generate the new plant. I mean, it's totally new plant at all. because the gene is already changed, something like that. And also allow them to move, be greatly reduce the chance of transmitting disease, and because in control condition, because if you plant at the outside, you need to alert the disease. You need to know not only human and animal got disease, plant also got disease. They have the chance to get the infection, and also the pest. Maybe insect. You need to worry about insect, and then you don't need to worry about the pathogen, which virus, something like that. Also, production of plant from seed that otherwise that have low chance of the germinating and growing. Yeah. So sometimes some plants you require you need to grow that plant is take a very long time, but using tissue culture you shorten the time, and also clean the particular plants of virus lah, so that clean stock lah. So yeah, so this is some of the advantages of the tissue culture lah. So this is a new topic. So normally this topic is the topic normally is entering university lah. And for those who are very interesting in the tissue culture lah. Yeah, okay. Okay, the third big topic is seed germination. Okay, so this topic actually is what I say I will continue from last chapter, right? Because one of the function of the plant hormone called gibberellin, they play a major function in seed germination. So at that chapter, I not touch it. So for the because I touched this in this topic, because in the syllabus you need to learn about the mechanism of seed germination in this chapter. Okay. Okay, so this is the overall process. Okay, so you already form the seed. From the embryonic development of the seed of the flowering plant, all the process. Okay, you already form the seed. Okay, you want to grow the plant, you need to germinate the plant first. You need to let the plant have the root, have the shoot first, then it can grow. So how do you germinate the seed? So this is the mechanism. So first of all, when you want to germinate the seed, you need a water first. Water is very important. Water is very important. So this one is initiated, we call as water imbibition. Imbibition process, which your seed will absorb the water. Water cause the seed to sweat and break down the seed cord, the outer surface. So once the seed cord is already break down, it was starting to germinate, and also the to wash out all the abscisic acid, because one of the function of the abscisic acid is to promote seed dormancy, meaning that to prevent the seed germination occur. So abscisic acid is highly concentration in the seed, so they use water to wash out, to reduce the concentration of abscisic acid in the seed. So this will trigger the change of the embryo, your embryo here, embryo starting to release the hormone gibberellin. Gibberellin. So this hormone gibberellin will diffuse into the alveolar layer here. You see here in this alveolar layer is more inner part, not the most outer part. The outer part is seed cord. It is at the inner under the seed cord. So this is the alveolar layer. You need to know alveolar layer. Okay, so once the gibberellin diffuse into the alveolar layer, it was starting to switch on the gene on synthesis specified protein in the alveolar layer. So the alveolar layer will secrete the hydrolytic enzyme, especially alpha amylase. Most important thing is alpha amylase. So what does this enzyme happen? This enzyme will diffuse into the endosperm. You need to know endosperm that form from the fertilisation, 3N, right? 3N. So this endosperm is very rich in the nutrient. So the enzyme alpha amylase will diffuse into the endosperm. So the starch that's stored in the endosperm will be hydrolyzed by alpha amylase. Alpha amylase to maltose lah. And then after that, maltose is hydrolyzed by maltase to glucose lah. So the glucose will act as the basic nutrient source absorbed by the embryo. So the embryo will use this all nutrient for cellular respiration. So apart from the alpha amylase, they also can produce proteins, can also produce lipase, but the most important thing that you need to write in examination is alpha amylase. So it will produce the sugar so that the embryo can take out this sugar for basic cellular respiration. And when the germination is completed, when the vesicle is emerged, followed by plumber. So that's all about the mechanism of seed germination. So actually it's very easy lah. And also came out many years already in essay lah. So this is a very easy topic. Also the factor affect the seed germination. Of course water. Water is the most important thing one. Also oxygen. Oxygen also can affect temperature, light, and pH of the soil. All that also can affect the seed germination. Okay, so the last topic is the growth curve. Growth. So based on the syllabus, there are growth curve. So there are 3 types of the growth curve that you need to learn. Okay, the first one is exotic growth curve. So you can see here this exotic growth curve. So what is the So for growth curve, you need to know the y-axis and x-axis. What is the y-axis present? X-axis present. So x-axis is time. Any time, any measurement time. Maybe years, maybe month, maybe minutes, maybe 10 years, months, or hour, all that. Y-axis is the parameters. Any type of parameter, including length, weight, height, dry mass or dry mass. So dry mass is for plant. Normally is for plant. So normally exotic growth curve is sigmoid curve shape to determine the growth of the organism over time by using the parameters. So it is to along the time what is your parameter growth? Is it growth or not? So normally is a sigmoid growth shape because when the time goes, the organism must grow right. The size of the organism must grow. So this is the exotic growth curve. Okay. After that, there are another curve we call as exotic growth rate curve. Okay, they add on the rate. Okay, so this means that you need to do a calculation, something of calculation, growth rate. So provide the growth rate is determined by the change in specific parameter over time, meaning that your x-axis still in time, but y-axis already changed, meaning it is changed in parameter. Meaning that, for example, maybe you use 3 years minus 2 years. If minus 2 years, any change or not? The change here, this first label is 3 years. 3 years. The change between the second year and first year, between third year and second year, between fourth year and third year, between fifth year and fourth year, like that lah. So normally you can see the bell-shaped curve like that lah. So normally this is a bell-shaped curve. So this is a growth rate curve. Okay. Finally, this is a relative growth rate curve. Okay, you can see here the graph is like that. The curve is like that. Okay, so why the curve is something like that? So you need to know the x-axis of y-axis first. So the x-axis still the time. Yeah, still the time. But y-axis differ. It provides the variation in growth rate over time, which reflect growth efficiency, defined as the rate of biomass accumulation relative to the initial size of the organism. Meaning that you need to do a calculation based on the initial size of the organism. So first one is the initial size one. And then after that, you need to minus until it reaches zero. So this is recalled as relative growth rate curve. Okay. So what coming out in examination, normal got 1 year, they're coming out in the graph, structural question, and then you need to draw a curve. Not pull out only, you need to draw. They already give you a graph paper, and then you need to do a calculation, and then want to find out the growth rate curve lah. They provide the exotic growth curve statistic, and then you need to find out the exotic growth rate curve and also relative growth rate curve. And then once you calculate it, right, you need to draw it down. 4 marks if not mistaken. Yeah. So that's all. So in exam mention, please be alert. Growth curve and growth pattern, they are different. The syllabus are separated. Growth pattern is growth pattern. Growth curve is growth curve. They are not same. Please don't say that when the question asks growth pattern, then you write the growth curve. And when the question asks growth curve, you write the growth pattern. Something like that. Please don't confuse it. There are two differences. Okay. So in growth pattern, first growth pattern is limited growth lah. So you can see here limited growth lah. So one of the example of the limited growth is human. So limited growth growth meaning that when they reach the sometime one of the period, it will become stationary. Your growth is stopped already, and after that it will die. The growth will be negative. So like human, so you can see here, red phase, the growth rate is very, very low. Normally it's in children, baby stage. And once you reach the puberty stage, normally it's secondary school, right? You do it secondary school, you see that growth is very fast. That's why that you in secondary stage, school, wow, you just 1 month not see only, wow, you already very tall. Because of exponential phase. Exponential phase, the growth rate is very high, it's the highest, and cells are actively divided and elongated. So the size of the organism of this at this stage is increasing very high, significantly. After that, you will reach the stationary phase. So stationary phase meaning that you're already starting to still go growth, but the growth is already very slow and almost stop already. Or stop already and starting to reaching the maximum size already. Then after that, you will reach the maturation phase. So maturation phase, of course, you already cannot go anymore lah. Your growth rate is constant now. Your size, your growth cannot increase anymore. Normally this one is you already reached the university state. 24, 25 years old, you already graduated from university until you are until you old until from 60, 70 years old. So after that, you will go through the census phase. Census phase meaning that older phase elderly phase, meaning that you starting to your growth starting to become negative. The organism starting to aging. All the metabolic rate starting to decrease, your health status, all that is starting to decrease. And finally, it's death phase. So your growth is totally negative because you already die. The cells cannot function anymore. It's totally stopped. Your cells are stopped already. So limited growth in human. One of the example is human, but also many animal and almost all animal is limited growth. And this difference is that some of the animal lifespan is very long, like turtle, tortoise, all the animal they have very long lifespan, but long lifespan. Okay. Also for annual plants, some of the plant that only including in the primary growth plant, they are also limited growth. They cannot grow the whole lifespan one. So for the annual growth in limited growth in annual plant is very special that at the initial phase, you can see here, the growth is decreased at here. So you need to know why this decrease happens. So normally it's due to respiration of stored food in the seed. So this happening is that during seed germination. There is an initial mass decrease in seed germination. Why? Because during seed germination, they require to take large amount of nutrient from the endosperm, right? You will still remember. So when the nutrient is already taken up, of course, your mass growth is decreased lah. Your growth will decrease lah. After that, once after seed germination, then it will grow until it reach the stationary phase. After that, it will die. So normally this one is limit for annual plant. The plant that cannot go the secondary growth, they only have primary growth. So what is primary growth, secondary growth? I think this one you already learned in SPM lah. So although in STPM you not learn, but I hope you still remember. Okay. So another growth is unlimited. So this one is only happen in the secondary growth plant, which is very woody plant normally. Normally it's woody plant. So meaning that it is non-stop. Unless you have the human action or there are disaster, earthquake, tsunami, flood, or human do logging activities or deforestation. Aside from that, the plant growth along the lifespan. So So meaning that this growth curve consists of a very series of smaller or sigmoid curve. So you can see like this one. So meaning that this is unlimited lah. It can go until here. Beyond here. So the growth is unlimited. Unlimited. Okay. So another growth pattern is that allometric growth in human. Okay. So that's why you see here, just mentioned about that. During the fetus stage, when you see the diagram, ultrasound picture during pregnancy, right, mother pregnancy, you see the diagram, head is very big. The body is actually very small. Very small. So allometric growth in human, growth of the different organ of an organism occur at a different rate from the rest of the body. Meaning that the organ of the growth is different from the rest of the body. Meaning that at the beginning, the growth, the organ growth rate is very high. So that's why that you can see the head is very high. You can see this diagram. At the beginning stage, the head grow rapidly one. The other organ go very small. And then the least grow is the reproductive organ. At the beginning stage, your reproductive organ will not grow very little only. Once you reach the puberty age, your reproductive organ grow rapidly. No? And then for your lymphoid organ is go rapidly from birth to early adolescence, and then after and go as go beyond the percentage. This is the final. This 100 is the final size of the organ. Final size of the organ, meaning that after that, all the organ is limited at here. But lymphoid tissue, your size will be increased larger than here. Larger than final size one. But after that, you will decrease back. So that's why that at this point, during the secondary school, you are very rare to get a sick one, right? Or even you get a sick, just tomorrow you already good. You already dah sembuh lah. Already can jump, can walk anymore. But compared to like the baby, because baby, your lymphoid organ is very weak. So that's why that in baby stage or in adult stage or even the older stage, the immune system is very weak. In baby stage, the immune system is very weak because the maturation. Your lymphoid organ still not mature yet. But in older stage, because of already too long, aging. But in secondary stage, your immune system is very strong. So that's why even you are sick or even you have any problem, infection, just few days you already can jump, can walk already. But compared to adult like us, we sick, maybe we need to take 3-4 days, a very serious that infection. Yeah. So this is a allometric. Another one is isometric. So isometric is the growth of the organism. Organ is similar to the rest of the body, meaning that like a fish, you can see the fish, their growth of the organ and the size is totally similar. Not like the human. You will see the human, you can see the size, the growth of the size of the body and the organ are totally different. But for fish, they are totally different. So this is what we call as isometry. The size of the body of the organism will change along the time. Okay, the final one, intermittent growth. Okay, so intermittent growth, one of the most, all the example is insect. Insect, but because of the few years past year, they do not use insect. They use other type of animal like crab, like crab. Or another one is another one is the I think I already forget, but they got 1 year they use the crab for the so very easy is arthropoda. So what is arthropoda? So arthropoda is the syllabus actually you will learn in semester 3 lah. But organism under arthropoda, they will undergo intermittent growth. So one of the example is insect, and then crab also one another example, and then Also seafood, the seafood, like the lobster, oyster, not oyster, lobster, one of the lobster, and then another one is spider. Spider also one type of arthropoda. So in general, I just want to tell you that intermittent growth is the happen where the organism they will conduct a molting process. This adhesis adhesis, which they have all exoskeleton are replaced with a new exoskeleton. So in the growth of the arthropoda, like insect, it will show like this. Show like this. Meaning that when there are adhesis, there are molting process, the growth is constant. After that, once they have molting adhesis, the growth is suddenly increase. No time limit. They just once they already molting, the growth is suddenly increase, and then the growth constant again, adhesis occur again, and then growth suddenly increase. So this is what we call as adhesis. So, when the animal goes large enough, that is completely fill its hard exoskeleton, it must lose that skeleton by molting. So, meaning that they have the hard exoskeleton, but then they still can increase. The body still increase, so that you can see here, they go very slow. But once they already reach the limit, due to the exoskeleton, so they will break down the skeleton, which we call as molting, change the exoskeleton. And then the ADC stage between them is we call as insta. Okay. So in intermittent growth, ADC is for insect. So we were more focusing on metamorphosis. Okay. So what is metamorphosis? So metamorphosis is one type of the ADCs where it is a change in shape occur very suddenly, and the process of changing from one stage to the following stage in the insect. So we know that in insect, they have many stage. Like butterfly. We know that it has the butterfly. We know that it has 4 stage, right? It has 8 stage. You have larva stage, and then you have pupa stage, and you have adult stage. There are 4 stage. But some of the insect they do not have stage. They just egg, and then after that just sim. They just go increase their body size only. So this one is we call as incomplete metamorphosis. That one is we call as complete metamorphosis. Which the development for incomplete metamorphosis, which in development in certain insect, the young insect resemble the adult characteristic, just a smaller and have different body proportion only. Just like that. So in metamorphosis, you need to learn about the role of the hormone in metamorphosis. So more focusing in complete metamorphosis. So one of the major example is using butterfly. So this is a book from diagram from the Campbell. So you need to know their brain structure. So their brain, they have the 3 major structure you need to know. The first one is neurosecretory cell. Second one is corpora cardiaca, and then the third one is corpora alata. So you need to know their location. So neurosecretory cell is the upper part one. The corpora cardiaca is the middle, and then the corpora alata is the lower part. Okay, so what are these 3 brain structure play? So neurosecretory cell will produce the hormone we call as proterotropic hormone, PTTH. You need to remember it. In exam, please don't use short form. You only can use short form in essay when you spell first time in first sentence. You need to mention about full sentence first, full name first, then you write backward PTTH, and then afterward you can use PTTH. But in subjective question, please use full name, because subjective question just a short answer question. You cannot use short form. If you use short form, no marks. Especially if the question asks about the naming. If they ask about naming, confirm no short form. If you still Right short form, no marks given. So you need to memorize lah. So this hormone will be secreted by this secretory cell and will store at the corpora cardiaca. Cardiaca. Okay. After that, they will have the gland, another gland in the insect we call as parturition gland here. Okay. So this parturition gland will secrete the addison. When the secrete addison, when the PTTH is released out from the corpora cardiaca. When E is released out, it will stimulate the parturition gland to secrete the hormone we call as addison. So in this book they use addistable lah. Also can acceptable, but we are more acceptable for addison lah. ACDYSONE addison. Okay, so you need to know the function of addison. And then for corpora alata will secrete the hormone for called juvenile hormone. Okay, so these two hormones they are in relationship to each other. So what is these two hormone play? So addison. This addison hormone is to trigger adhesis process, molting process, and development of adult characteristic. Meaning that when this hormone is present, it will cause the insect change have the metamorphosis process like this and change the insect from one stage to adult characteristic. Like if in larva, meaning that this hormone will change the larva state into the pupa stage, and then this pupa state will change to the adult stage in this hormone. Okay, how about juvenile hormone? So you already say juvenile. Juvenile is the remaja, the teenagers. So meaning that this hormone is to suppress the metamorphosis process. So this addison induce metamorphosis. It induce both. It induce metamorphosis and also adhesis. But juvenile hormone suppress metamorphosis. So meaning that in the larva stage, high juvenile hormone is happening, is at here. So even though the addison is present, they only induce adhesis only, no metamorphosis. But when they want to change to pupa stage, when the GH hormone starting to decrease. Okay, when the GH hormone starting to decrease, low amount, addison still present, it will induce the induce the adhesis and also metamorphosis. So just very easy, addison induce metamorphosis and adhesis. So when you want to inhibit the metamorphosis, only juvenile hormone can inhibit it. So during larva stage, juvenile hormone is high concentration. When they want to reach the pupa stage, juvenile hormone starting to decrease. When it decrease, addison will not only induce molting, they will also induce metamorphosis. So when the develop into pupa and develop into adult, juvenile hormone is starting to decrease, especially in adult. Normally no juvenile hormone anymore. So that's all for this chapter. This chapter is very long chapter. There are 4 main topics at all. So hopefully this free class video can provide the free lesson to you guys to understand what you will learn in STPM Biology for this chapter, chapter 11, Reproduction, Development and Growth. So thanks for those who are hearing from beginning until end, and good luck for your Sem 2 lessons, Sem 2 journey, and also good luck for your coursework and experiment, and finally is your examination. So hopefully you can get your good marks in your examination, and thank you and bye.